Hello everyone, welcome back to eJournal Club, where we simplify cutting-edge reproductive pharmacology in just a few minutes. Today's featured article is titled Microfluidic Technology and Simulation Models in Studying Pharmacokinetics During Pregnancy, published in Frontiers in Pharmacology in 2023. In this episode, we'll explore how organ-on-chip technology and physiologically based pharmacokinetic modeling, or PBPK, are transforming the way we study drug disposition during pregnancy and why this could change the future of maternal and fetal medicine. Developing medications for pregnant women has always been a major scientific challenge. Because pregnant women are rarely included in clinical trials, we have limited information about how most drugs behave during pregnancy. Researchers have traditionally relied on animal models or placental perfusion studies. However, animal physiology differs significantly from humans, and placental perfusion experiments only evaluate the placenta after delivery. Neither model fully captures the complexity of pregnancy. So, how can we create a model that better represents what actually happens inside the human body? That is exactly what this study set out to address. Before discussing the technology, let's first understand the feto-maternal interface. This is the highly specialized biological boundary where maternal tissues communicate with fetal tissues throughout pregnancy. It is much more than just the placenta. The placental interface contains syncytiotrophoblasts, cytotrophoblasts, and fetal endothelial cells. The fetal membrane interface consists of the maternal decidua, chorion trophoblast cells, amnion mesenchymal cells, and amnion epithelial cells.
Together, these 7 interconnected cell layers regulate immune signaling, inflammation, nutrient transport, waste removal, and most importantly, drug movement between the mother and the developing fetus.
Understanding how drugs cross this interface is essential for designing medications that are effective for the mother while remaining safe for the baby. To recreate this complex biological system, the researchers developed a feto-maternal interface placenta organ-on-chip, or FMi-PLA-OOC. Instead of studying each tissue separately, they cultured all 7 cell types inside interconnected microfluidic chambers. These chambers are connected by tiny fluid channels that allow continuous communication between tissues closely mimicking human pregnancy. Next, they selected pravastatin as the model drug. Pravastatin was introduced into the maternal decidual chamber. Samples were collected after 2, 4, and 8 hours from each compartment. Drug concentrations and metabolites were then quantified using mass spectroscopy. At the same time, the team built a pregnancy-specific PBPK model using Gastroplus software to computationally predict maternal and fetal drug exposure. This allowed direct comparison between experimental observations and computer simulations. The results were impressive. As time progressed, pravastatin gradually moved across the interconnected cellular layers from the maternal side toward the fetal compartments. Drug concentrations increased in downstream tissues over time. The investigators also detected pravastatin metabolites in multiple compartments. This finding revealed that drug metabolism is not limited to the placenta. Instead, several tissues within the interface actively contribute to drug biotransformation. The researchers then compared their experimental findings with computational predictions and previously published human data. The agreement was remarkably close. The organ-on-chip predicted approximately 18% fetal drug transfer. The PBPK model predicted approximately 16%. Human placental perfusion studies reported approximately 18% as well. In contrast, traditional mouse models predicted nearly 33% fetal transfer, substantially overestimating fetal exposure. These findings demonstrate that combining human-based organ-on-chip technology with mechanistic PBPK modeling provides a much more accurate representation of pregnancy pharmacokinetics than conventional animal studies. This study represents an important step toward the future of pregnancy drug development. Rather than relying exclusively on animal experiments, researchers can now integrate microfluidic systems with computational PBPK modeling. These technologies also support the growing movement toward new approach methodologies, or NAMs, which aim to reduce animal testing while improving the prediction of human biology. As regulatory agencies increasingly recognize these approaches, platforms like these may become essential tools. So, here's a question for you. Could organ-on-chip technology eventually replace animal models in pregnancy research? And should PBPK modeling become a standard part of pregnancy drug development? Share your thoughts in the comments below. If you enjoyed this episode, don't forget to like, subscribe, and join the discussion on Substack. Thank you for watching eJournal Club, where we make reproductive pharmacology simple, engaging, and accessible. I'll see you in the next episode.